To understand life is more than to define it. And to define life is presently a questionable endeavor. I should like to advocate my own fundamental conviction and a means of circumventing the definition problem or postponing it until we know better. A conviction which is best summarized by Richard Feynman's famous, "What I cannot create, I do not understand." We can ultimately only understand what life is once we are able to create or recreate it ourselves, experimentally from clearly inanimate to clearly animate. It may be that we would then witness a life form somewhat different from our own. Possibly, and quite likely, after repeating the experiments, we would perceive even more divergence. And yet, we could watch synthetic lives emerge and evolve in complexity. We could deduce from these observations our comprehension of life that appeared on Earth some 3.5 to 3.9 thousand million years ago. Besides, we could convince many more contemporaries that life emerges without any external help, any other help than our own at least. If we humans can do it, create life from the inanimate, nature, unlike building refrigerators or cars or aeroplanes, can do it without us. There is a caveat that is recurrently brought up during discussions on what nature can do and what humans cannot. Time is a crucial factor. Many say nature disposed of millions of years, even today some say, to produce life. We scientists do not. My immediate, somewhat agitated answer to this is, how do we know that nature needed such a long time to produce the first living matter? I don't. I cannot remember. I'm too young to have witnessed the origin of life. There can be no proof for the million years assumption. It must say an unscientific statement. It is not falsifiable. We may, however, quite safely assume that evolution of our ecosphere has taken millions of years, not of life per se. For the origin, we can reflect upon a reasonable minimal time frame that must have been at disposal at this particular place and time, when now and again chemical conditions on primordial Earth were such that for limited periods of time populations of compartmented matter appeared, consumed energy, consumed exogenous material, free energy, grew in population size, changed its immediate environment by excreting waste, and so on. Would we be present and watch it happening? Many of us would call it alive, animate, behaving as if it had a purpose, teleonomic, until, for some lack or other, side reactions, the phenomenon would have vanished, degraded, fallen into its thermodynamic pit hole, dematerialized, died. Such events might have taken place many times, in many places, during the same or different stages of primordial Earth. They need neither have been homogeneous in the sense of similar from case to case, nor have they led to any continuity, monotony, other than vanishing and reappearing for the same repeated physical-chemical reasons, and in the absence of any systemic memory, being based on replication and inheritance. A single appear and vanish cycle could hardly endure and evolve over millions of years. The chemical persistence of the organic molecules, let us avoid here the term lifetime, the persistence needed for keeping the system away from thermodynamic equilibrium is much shorter. We are not considering the persistence of possibly simple and thermodynamically quite stable building blocks like purine bases or amino acids, but of supramolecular assemblies based on hydrogen bonds and other chemically weak interactions. So, a number of highly respectable colleagues would not quite describe the above phenomenon as animate. One axiomatic requisite for matter to behave alive must be a replication-based systemic memory, thus include a templating device that would allow for the heritage of much of the acquired systemic knowledge. Also mandatory is the ability of the system to produce endogenously some of its needed high-energy compounds to replace exogenous compounds, to become efficient in energy gathering and thus more independent from the environment. So, how to experiment on the creation of synthetic cells? My methodological conviction is in full agreement with what Adi Pross describes in his new book as hierarchical reduction. The idea being that phenomena at one hierarchical level can only be explained using concepts taken from a lower hierarchical level. You can only explain biogenesis by using the concepts of chemical and physical sciences. Staying at the biological level of complexity will make it impossible to ultimately comprehend life's emergence, and even more so to reinitiate life from the entirely inanimate. This means, for example, that we need to find chemical conditions for replication to actually emerge and evolve, rather than to start off with a designed replication system right away. My other methodological credo is the following. In our attempts to set up experiments that are aiming at the observation of lifelike behavior in initially inanimate chemical systems, we should better avoid creating metaphors of life, that is, mere images, prone to be mistaken for a system that is truly alive over a period of time. So, if a colleague constructed a chemical system that contained a compartmented and truly exponential replicator, whose function depended on a, say, designed model metabolism, the system persisted unchanged unless it grew in population size, as long as sufficient energy were provided. My short answer would be, ceci n'est pas une Bible. My longer answer, an essential attribute of any living chemical system, actually irrespective of composition, size, complexity or location, must be the evolvability of its diversity in time. If the described model system, the chemical model system, represented a certain stage of complexity of life, if it were kept far off its thermodynamic equilibrium during that period of time, but without ever changing into any other slightly different, persistent off-equilibrium system. Just changing into thermodynamic degradation products, then it were not living after all. It would be just a metaphor, an image, if you like. Perhaps a beautiful metaphor or photographic image of life. Metaphors can be cited, can be used for insightful descriptions, for learning, for teaching. Images can be framed, carried around, be useful for remembering. But on their own, metaphors or images are never able to walk out of the beaker, that is, to act autonomously, as if on purpose, let alone learn how to play the violin, evolve. In this sense, refrigerators are metaphors for warm-blooded animals that live in hot areas, desert hedgehogs, dingoes, humans. Cars are metaphors for fast-running animals, like cheetahs, hares, horses, or humans too. And aeroplanes are metaphors for actively flying animals, bats, birds, dragonflies, mosquitoes. Briefly, we cannot aim at a certain wanted and observed defined result that would well represent a living system instead of actually being one. Almost by definition, we cannot know precisely what the experimental successful outcome will be, should be, or look alike, because the successful outcome must inherently diverge in time and variety space. So, the construction of synthetic living cells has become a declared goal of a growing community of chemists, systems chemists, for example. Attempts to experimentally reconstruct presumably ancient macromolecular self-organizations must be a valid approach to follow this goal. A deeper knowledge about the spontaneous supramolecular organization of macromolecules on primordial Earth may eventually serve to comprehend the emergence of a dynamic complex chemical system of any composition, natural, semi-natural, or wholly synthetic. What we can do initially, according to my feeling, in the first experiments, is to opt for what I call a prudent compromise, to stay reasonably close to the molecular composition of the building blocks of current biological systems. A new kind of in vitro selection experiment is required, being held as far off as thermodynamic equilibrium as experimentally feasible, and facing a plethora of dynamic, ill-defined selection barriers instead of only one or two that seem chemically useful, like ligand binding, for example. Let us follow a set of rules. I should like them dogma 13. First, no biologically evolved information system, such as the universal genetic code, should be implicit in the starting compounds or starting conditions. Second, the emergence of a de novo genetic code is actually ultimately sought for. Third, the highest degree of information to embark on in the initial chemical system can be chemically or stereochemically specific catalysts. Specific catalysis being a form of chemical information. Fourth, prohibited is the use of any biologically evolved products, such as known enzymes, biosynthetic metabolites, for example, secondary metabolites, cholesterol, sphingolipids, and the like. 5. Except for those that are likely to have formed abiotically, so certain amino acids, hydroxy acids, long-chain fatty acids, long-chain alcohols, carbohydrates, polyols, simple amides, and heterocycles, nucleosides, nucleotides, and the oligomers and conjugates of all those listed. 6th, if we start out homochiral, this already would be a prudent compromise. So, once the compromise experiments grow to be reasonably successful, one should go back and restart heterochirally. The long-term goal is to find feeding conditions for an energy-consuming chemical system to become dynamic, more precisely, dynamically sustained over a prolonged period of time, during which a more or less constant turnover of its constituent is maintained. Its macromolecular constituents are synthetic nucleic acids, synthetic peptides, and carbohydrates, all compartmented at or into lipidic vesicles that ultimately shall be replicating to give more vesicles. At least a part of the contained nucleic acids should replicate as well, that is, multiply their nucleotide sequences and redistribute them into the daughter vesicles. In order to achieve this, and in view of the methodological credos described before, I think the best way is to provide conditions that seem favorable, if not compulsory, for the spontaneous formation of some kind of translation system between the nucleic acids and the peptides. The highest imaginable success would be the discovery of a or several de novo genetic codes. The decades-long research on enzyme-free autocatalytic and cross-catalytic nucleic acid amplification, particularly the search for truly exponential replicators growing in population size exponentially, is, in my opinion, in my humble opinion, pointing to the non-existence of any primordial RNA world. The foremost argument in favor of RNA self-replication in a primordial RNA world is nature alleged to be parsimonious, that is, economic, sparing, cost-conscious. Therefore, one follows, nature first generated an animate system where function, catalysis, and replication were combined in not more than one molecular class, RNA. This is not convincing anymore. Nature is not necessarily parsimonious. Nature does whatever works by letting evolvable autonomous agents explore the adjacent possible. If we agree upon an inhomogeneous messiness of the spontaneously available inanimate environment prevailing on primordial Earth, as opposed to the selected conditions maintained in a clean scientific laboratory, then an animate single molecular class world of any kind was hardly adjacent. Whatever was adjacent possible for becoming the cradle of life, out of a mixture and flow of then available prebiotic compounds, was very likely to be shouldered by several, if not many, actually quite different molecular classes at a time. by those that get spontaneously organized in functionizable macromolecular assemblies bearing a hollow interior, so lipidic amplifiers, by those that are modular and offer rigid polar three-dimensional fragments that replace solvation water molecules at low entropic cost, carbohydrates, by those that are highly modular and extremely variety-rich in their catalytic and structural properties, amino acids, peptides, proteins, and last but of course not at all least, by those that are perfectly water-soluble at virtually any molecular length, nucleic acid, irrespective of the nucleic acid's actual backbone conformation. The two-dimensional flat hydrogen donor acceptor patterns can be read as templates at relatively low entropic cost. These features make them ideal for the copying of any nucleotide sequence through twice loss-free doubling of the amounts of the molecules, which eventually results in exponential population growth. Nonetheless, the discovery of exponential self-replicators or cross-replicators is stubbornly withstanding any general resolution. In spite of long and intense research efforts on RNA and a number of related nucleic acids. The reason for this is largely known in principle. Both product inhibition and side reactions cause over generations a steady loss in doubling efficiency. However, any non-nucleotidic yet easily evolvable molecular device being able to perform loss-free nucleic acid doubling may carry out, catalyze, exponential nucleic acid replication. Modern nature knows well how to loss-free double nucleic acids through the use of protein enzymes, polymerases, with the help of activating compounds, ATP, and chelating ions, magnesium. It seems a much longer way to evolve from a mixture of different compound classes, an inanimate system that essentially relies on a single performer, as in the RNA world. So, from a strictly evolutionary point of view, where the initial condition is some mixture of abiotic molecules submerged in a flow of higher energy prebiotic compounds, I think we could reasonably and convincingly ask, why bother using uniquely RNA for all functions, including loss-free doubling, if a mixture of 3 to 4 available compound classes can do a better job? Whatever works better at the moment will happen first. Whatever happens first will persist until it is outcompeted. This is where translation comes in. This is the ultimate reason for evolving a reliable and recurrent production of proteins for which a genetic code is prerequisite. Any genetic code will couple easily templating nucleic acids, RNA, with their loss-free doubling, replication, through protein enzymes. The reproducible production of useful proteins, as opposed to any peptides or proteins being available by mere chance, must have emerged within the relatively short time frame of a single appear and vanish cycle mentioned earlier. A chemical system unable to reliably replicate all its molecular constituents, thus remaining heavily dependent on a steady exogenous supply, but able to produce proteins that catalyze loss-free RNA replication, this is the bottleneck nature had to squeeze through. Whether to name such a chemical system capable of evolving a genetic code alive or not yet alive is academic. But according to process principle of hierarchical reduction, the emergence of a genetic code operating in an inanimate chemical system must precede the upheaval in complexity to a fully replicating chemical system. So, in my opinion, wise experiments will aim at the emergence and evolution of a translation system implying proteins instead of the replication of nucleic acids without proteins. A genetic code is based on actually two recognition principles, or else it will not work. Today's primary genetic code is the recognition and correct distinction of codons by anticodons, which is a pure templating quality between messenger RNA and transfer RNA. It is carried out by an ancient ribozyme, the ribosome. Today's secondary genetic code is the recognition and correct distinction of amino acids and cognate transfer RNAs by proteins. Amino acid transfer RNA synthetases are the enzymes that guarantee for a correctly translated code. Primordial functional fragments of both ribosomes and of amino acid transfer RNA synthetases may have co-evolved. Experiments to recreate translation are the key for a chemical system to become animate. The question now becomes, if we know where nature was heading for, translate to replicate, where did she begin with? What were the pathways that allowed for stepping close to this adjacent possible? What physical-chemical feature would keep proteins and RNA in close contact to one another? And how to compartment RNA? Natural liposomes are compartments ideal for the growth and harboring of information carriers, of useful metabolites, and for their own reproduction. The bilayer surface of lipidic vesicles may have served since long for the anchoring and enrichment of macromolecules, which sooner or later have been internalized and retained in the interior of the vesicles. Surface-bound internalized hereditary molecules, like RNA for example, could then grow in population size and length more rapidly, safer and more reliably. So an obvious challenge for the deliberate creation of a dynamic off-equilibrium system that is expected to show signs of life, that is, sustained population growth of certain, not all, initially present individuals, is the concentration and compartmentation of nucleic acids at and into the lipidic vesicles. Replicating primordial cells, we call them natural protocells, are expected to have emerged from chemical systems that were exploring the affinity between natural nucleic acids and natural lipidic vesicles. They hate each other, usually. By means of a molecular anchoring device. Useful devices should be sufficiently abundant, composed of prebiotically implausible precursors, offer finely tunable properties without being highly evolved, thus enhancing the potential for the system to become self-evolvable. Lipidic anchoring compounds do bear some potential of evolvability, but In the absence of enzymes that catalyze the formation and breaking of carbon-carbon bonds, far less so than the evolvability of the highly modular compound class of peptides, where the hydrolysis and formation of carbon-nitrogen bonds seems more amenable to abiotic catalysis. In prebiotic times, activated, meaning particularly energy-rich compounds, mineral or organic anhydrides of some sort, were likely available during geologically active periods on Earth. Ultimately, it is owing to such anhydrides that the abiotic formation of organic macromolecules of the oligomer type may be explained. Saccharides, oligosaccharides, oligopeptides, nucleic acids, lipids, fats are all products that form upon dehydration of their monomeric precursors or fragments thereof. Aldehydes, ketones, amino acids or amines, phosphoric acids, fatty acids, and alcohols or polyols react to give, respectively, acetals, carbohydrates, amides, peptides, esters, lipids, nucleic acids. Chemically, it's dehydration. However, the chemical selectivity of those abiotic condensing anhydrides is very limited. Consequently, different chemical families of mixed chimeric condensation products, such as peptidic nucleic acids or lipidic sugars, etc., were being combinatorially produced and intermingled as well. The assembly and propagation of protocells could only be driven by quite simple and straightforward physical forces such as solubility, amphiphilicity, osmotic, pH, or ionic strength gradients. So, chimeric amphiphilic macromolecules are therefore likely to have played a crucial role in the assembly and organization and development of such early protocellular structures. Here we shall argue that hydrophobic peptides composed of prebiotically available alpha amino acids are likely to have functioned as natural and readily evolvable anchoring devices for nucleic acids, for example RNA. Such peptides can be reversibly linked to RNA by means of prebiotically plausible condensing agents without the requirement for specific catalysts such as ribozymes. When amphiphilic peptide RNA conjugates are attracted to lipidic vesicles, this means barely soluble, otherwise aggregated or precipitated hydrophobic peptides are being mobilized with the help of RNA, thus made more readily available for chemical systems. At the same time, highly soluble RNA are being immobilized with the help of the peptides, thus saved from rapid dilution out of the system. This is a mutual advantage for two quite different compound classes. As a result, a fraction of those randomly generated amphiphilic peptide RNA conjugates are expected to possess sufficient transmembrane properties to internalize into the interior volume of the lipidic vesicles by virtue of any feasible membrane crossing mechanism. Once inside, the peptide anchors may hydrolyze, and the RNA may grow in length and population size in the presence of chemically activated nucleotides. This utility should initiate upon continuous feeding a selective process that enhances the sustainability of the functionalized vesicles with respect to the non-functionalized vesicles. This owing to a better import-export control across the lipidic membranes reminiscent of of modern transmembrane proteins. A logical consequence on a longer term could be the emergence inside the vesicles of a translation system and genetic code, as we know it from modern organisms. Once experimental conditions are found that allow for an in vitro evolution of chemical systems that start with amino acids, peptides, nucleotides, sugars and lipids, but no predisposed genetically encoded enzymes, the de novo emergence of synthetic living cells should be in close reach. In conclusion, we aim at exploring through experimental evolutionary reconstruction, based on the rules of Dogma 13, the process of how nature learns to take advantage of physical-chemical properties of any available peptide that might spontaneously assemble from simple alpha amino acids that themselves might form spontaneously, as in the Miller-Urey experiment, and be reversibly linked to RNA that in principle may form spontaneously, with the effect of compartmenting RNA molecules into the interior of dynamic lipidic vesicles that self-organize spontaneously, ideally from spontaneously formed fatty acids and glycerol, which, I admit, is only a final goal, as so far. We will first use commercial lipids. We are planning to exploit experimentally the pathway from a moderately complex chemical system towards organized complexity, adaptability, eventually to evolvability, with the goal of giving a proof of concept that such chemical systems might become self-evolvable. So for this meeting, of course, we ask ourselves, why RNA? More 2 decades of research on many different kinds of nucleic acids, synthetic or semi-synthetic structural analogues of DNA and RNA, have convincingly shown that an astonishingly high number of such oligomers or polymers do base pair with one another. Many nucleic acid variants can be used for templating, thus be potentially exploitable for faithful nucleic acid doubling, be it through polymerization, ligation, or a combination of both. This is good news and bad. The base pairing potential of such nucleic acids may be promiscuously high. It makes us keep asking again and again, why was RNA chosen for inheritable replication? Why not one of the other nucleic acid candidates? Because RNA replicates best is hardly a good answer anymore after those years of research. Because RNA base pairs best is even worse an answer. It's not true. Because natural proteins can polymerize RNA best is probably wrong as well. Proteins are just too good in adapting to any kind of useful, exploitable molecular recognition and catalysis. However, RNA molecules are expected to outclass other nucleic acids in synthesizing peptides from chemically activated amino acids. By virtue of their uniquely unhindered vicinal 2'3' cis-diol terminus, a feature that still is the basis of modern ribosomal protein biosynthesis. No other furanosyl nucleotide offers 2 free cis-hydroxy groups at the terminus of the oligomer. The 3' terminus of RNA is catalytically highly competent, It carries out conformationally defined internal movements, allowing the hydroxyls to switch between equatorial and axial orientations at low energetic costs. Pyranosyls, for example, are too rigid for this. As for lixols, for those who think about lixols, that also contains two-terminal cis-hydroxy groups, either one is sterically too hindered in alpha or beta lixofuranosyl nucleotides. So the working hypothesis is because RNA makes proteins best. That's the reason why we are living in an RNA world now, which emphasizes again the importance of finding chemical systems that will kickstart translation off its yet unknown grounds. The vision is suitably nurtured vesicle populations of appropriate lipid compositions are expected to evolve at the same time protoforms of ribozymic ribosomes and proteic amino acid transfer RNA synthetases and nucleoside triphosphate synthetases, thus develop simultaneously a persistent, if primitive, primary and secondary genetic code. The components needed for a persistent systemic memory, that is, a genetic replication machinery based on primordial nucleic acid polymerases, fatty acid synthetases, ribonucleotide deoxygenases, will follow rather than precede translation. This means that the experimental testing of evolvable chemical systems must be held under full feeding conditions for a long enough time period. These conditions will permanently, or at least recurrently, provide the system with small to medium-sized molecular precursors and chemical energy to allow for the emergence of the first compartmented and functional proteic enzymes. I guess Dogma 13 should help to achieve this within a scientist's lifetime. This work would never have been possible without the devoted action of a number of collaborators. They're all listed on the left side in a chronological order. That's the experimental part, which I have talked about. But I must mention colleagues with whom I discussed through the years about all these things. The most important for me are Günther von Strazewski, Urs Schattenmarie, Luigi Luisi, Albert Eschenmoser, Hans Kuhn, Stuart Kaufmann, and Ali Prost. All those discussions have molded on my way of thinking in what it has become now. So my deepest thanks to all these scientists. And the financial supports from the European institutions are gratefully acknowledged.